What's up, you guys? Welcome to part 2 of our lab demonstration. Remember the setup that we used for the dialysis tubing last lesson? This had the starch and the water on the inside of the dialysis tubing, but the one that you guys did not see me put together was this one right here. So this is just sugar water, glucose water, and I placed it into this beaker that had pure water. Here on the left I have iodine and this solution that's called Benedict's qualitative solution. Both iodine and Benedict's are what we call indicators. In chemistry, we can use these and we can add them to certain solutions to identify or indicate if other certain specific types of molecules are present. Iodine, it will turn from like an orange color to black if there is starch present in a solution. Benedict's will actually turn from a nice blue color to this green or even a yellowish orange or even red in color if there is glucose present in the solution that we add it to. If we look at water and iodine, it's got this nice burnt orange color to it. And I did the exact same thing with water and Benedict's solution. It gives us this nice light blue color, okay? So over here I've got the glucose in water solution. And what I'm going to do is I'm going to be using microcentrifuge tubes to take a sample of the water that's outside of the dialysis tube as well as the water that's on the inside of the dialysis tube, okay? So first things first, I need to get a sample of the outside water. I'm going to take it and just add about 1 milliliter. Anytime I use a pipette, I got to make sure that I put it back here in my used pipette cup. I'm going to cap this. So this is the outside of the dialysis tube with glucose and water. But then I got to get a sample of the water that's on the inside of the dialysis tube. Really take a pair of scissors. Go ahead and cut my dialysis tubing. And I need to take my pipette and I'm going to get a sample of the liquid from the inside of this dialysis tubing. And I'm going to place it into another microcentrifuge tube. And now I have a sample from the inside of my dialysis tube. Let's go ahead and test this sample using the Benedict's solution indicator. 1, 2, 3, 4, 5. Now let's sample from the inside of the dialysis tube. 5 drops of Benedict's to this one. 1, 2, 3, 4, 5. And I have my sample from the inside of the dialysis tube that had glucose and water on the inside. I'm going to now do a sample from the outside of my dialysis tube that contained the starch and water solution. So this is the one that you guys saw me put together in part number 1. I'm going to take a sample of the water from out here on the outside of the tube. I'm just going to add 1 milliliter, fill it up right there. I'm going to cap this, cut open to get to the inside of the dialysis tube. Got another. Oh, I'm stuck. Oh no. Uh-oh, you guys, this is why I need you here. I'm going to get my sample before I lose everything from the inside of the dialysis tube that had the starch and the water on the inside. I'm going to add it to another brand new microcentrifuge tube. Put my used pipette back. I'm going to cap this. That was a trip. Now I'm going to use some iodine, the indicator for starch, and we're going to see if there was any starch on the outside of the dialysis tube. 1, 2, 3, 4, 5. Set it here. A sample from the inside of my dialysis tube. 1, 2, 3, 4, 5. I'm going to go ahead and cap that one. Okay. Iodine plus water, just regular pure water. Iodine plus water is that nice burnt orange color. And we know that iodine can turn from orange to black if there is starch present. So on the outside of the dialysis tube, the one that had starch on the inside and water, this is what it looked like when I took a sample of the water outside of the dialysis tubing. But now check this out. Iodine is indicating that there is absolutely starch present on the inside of the dialysis tube. That's why it turned to black, because iodine turns from orange to black if there is starch present. Okay? Okay, let's talk about the water just with the Benedict's solution. This is what water looks like when we mix it with Benedict's solution, just pure water. And remember, Benedict's turns from like a blue to a green or an orange or a red if there is glucose present. The very final step of this lab is to boil some water, and then I put that water right here into this beaker. And in order to fully activate the Benedict's solution to tell if there is glucose on the outside of the dialysis tube and on the inside of the dialysis tube, is I need to go ahead and set these two microcentrifuge tubes into this beaker of water. What I'm going to do is I'm going to let these bad boys sit for about 5 minutes. Ooh, nice. We can already see something happening. Again, let me go up and over here. Remember that Benedict's solution, it will turn from this nice bluish color to a green or an orange or a red if there is glucose present. Okay, it has been 5 minutes. The microcentrifuge tube that you see on the left, that is the solution from the outside of the dialysis tube that had the glucose and water. We added the Benedict's, and then in order to activate the Benedict's solution, we put it into this hot water, and it has definitely changed color. Remember, this is what it looked like when it was just the regular old water and the Benedict's solution. And then when I took a sample of the water from the, or I should say the solution from the inside of the dialysis tube, and this is again the one that I started with by putting glucose water into the inside of the dialysis tube. After adding Benedict's, activating the Benedict's, this is what we see over here on the right, okay? Again, this would just be water and Benedict's solution. This is definitely this, oops, this one has no sugar present in it, no glucose present in it. So if you had to take a wild guess about these two, what do you think? Alrighty. And that is the end of lab part number 2. Have some great discussions as a class.